Very good day to the viewers. Today we are going to discuss about ketogenesis. First, we'll see what are ketone bodies. The compounds namely acetone, acetoacetate and beta hydroxybutyrate are known as ketone bodies. Only the first two are true ketones, while beta hydroxybutyrate does not possess a keto group. Ketone bodies are water soluble and energy yielding. Acetone, however, is an exception since it cannot be metabolized. Then ketogenesis. The synthesis of ketone bodies occurs in the liver. The enzymes for ketone body synthesis are located in the mitochondrial matrix. Acetyl-CoA formed by oxidation of fatty acids, pyruvate or some amino acids is the precursor for ketone bodies. Ketogenesis occurs through the following reactions. In the first step, 2 moles of acetyl-CoA condense to form acetoacetyl-CoA. This reaction is catalyzed by ketothiolase, an enzyme involved in the final step of beta oxidation. Hence, acetoacetate synthesis is appropriately regarded as the reversal of thiolase reaction of fatty acid oxidation. In the second step, acetoacetyl-CoA combines with another molecule of acetyl-CoA to produce beta-hydroxy beta-methyl glutaryl-CoA, that is HMG-CoA. HMG-CoA synthase catalyzing this reaction regulates the synthesis of ketone bodies. And in the third step, HMG-CoA lyase cleaves HMG-CoA to produce acetoacetate and acetyl-CoA. In the fourth step, acetoacetate can undergo spontaneous decarboxylation to form acetone. In the fifth step, acetoacetate can be reduced by a dehydrogenase to form beta-hydroxybutyrate. The carbon skeleton of some ketogenic amino acids is degraded to acetoacetate or acetyl-CoA. Example, leucine, lysine and phenylalanine. Utilization of ketone bodies. The ketone bodies, being water soluble, are easily transported from the liver to various tissues. The two ketone bodies, that is acetoacetate and beta hydroxybutyrate, serves as important source of energy for the peripheral tissues such as skeletal muscle, cardiac muscle, renal cortex, etc. The tissues which lack mitochondria, however, cannot utilize ketone bodies. The production of ketone bodies and their utilization become more significant when glucose is in short supply to the tissues, as observed in starvation and diabetes mellitus. During prolonged starvation, ketone bodies are the major fuel source for the brain and the other parts of central nervous system. It should be noted that the ability of the brain to utilize fatty acids for energy is very limited. The ketone bodies can meet 50 to 70% of the brain's energy needs. This is an adaptation for the survival of the organism during the periods of food deprivation. Now we'll see overproduction of ketone bodies. In normal individuals, there is a constant production of ketone bodies by liver and their utilization by extrahepatic tissues. The concentration of ketone bodies in blood is maintained around 1 mg per deciliter. Their excretion in urine is very low and undetectable by routine test. Ketoanemia, which means rise of ketone bodies in blood above normal level, is known as ketonemia. Ketonuria, when the blood level of ketone bodies rises above the renal threshold, they are excreted in urine and are called as ketonuria. Then ketosis. It is the accumulation of abnormal amount of ketone bodies in tissues and body fluids is termed as ketosis. It is most commonly associated with starvation and severe uncontrolled diabetes mellitus. Starvation is accompanied by increased degradation of fatty acids to meet the energy needs of the body. Diabetes mellitus is associated with insulin deficiency. This results in impaired carbohydrate metabolism and increased lipolysis, both of them ultimately leading to the accumulation of acetyl-CoA and its conversion to ketone bodies. In severe diabetes, the ketone body concentration in blood plasma may reach 100 mg per deciliter, and the urinary excretion may be as high as 500 mg per day. Thank you. Thank you for listening the video.